Hi, my name is Suhyule Kim, and today I'm excited to present my research titled Investigating the Role of Cold Water Therapy in Modulating Brain Cell Health and Antidepressant Gene Expression. This project was initially inspired by the increasing popularity of cold exposure therapies such as cold showers, ice baths, and cryotherapy on social media. Many people claim benefits like enhanced mental clarity, mood improvement, and reduced stress. But do these claims have a cellular or molecular basis, especially in terms of brain health? My research aims to explore this in the next few slides. Cold water therapy is gaining attention for its potential to boost immunity, speed up metabolism, reduce inflammation, and more recently, influence mental health. Scientifically, mental health conditions such as depression have been linked to decreased neuroplasticity, the brain's ability to reorganize and form new connections, and lower levels of CREB and BDNF. CREB, CAMP response element binding protein, is a transcription factor important for memory and mood regulation. BDNF, brain-derived neurotrophic factor, helps support the survival of existing neurons and encourages the growth of new neurons and synapses. So I asked the question, can cold exposure influence CREB and BDNF expression in brain cells, providing a possible neurobiological mechanism for the mental health benefits people report? To answer this, I designed a controlled in vitro experiment using human brain glial cells exposed to cold temperatures. For this study, I cultured A172 human glial brain cells and exposed them to cold water conditions, specifically 4 degrees Celsius, for 5, 15, 30, and 45 minutes. A control group was kept at normal conditions, placed at body temperature of 37 degrees Celsius. After exposure, I examined cell morphology using light microscopy, cell viability using Luna-FL cell counter, and gene expression using RT-PCR for two key markers, CREB and BDNF. All experiments were done in replicates to ensure consistency, and GAPDH was used as a housekeeping gene to normalize the RT-PCR results. I specifically chose glial cells because they play a key role in supporting neuronal function and are highly sensitive to environmental changes, making them ideal for this type of preliminary investigation. The first outcome I looked at was whether cold exposure damages brain cells or not. Here, you can see the morphological images of the cells at different time intervals under cold conditions and in the control. Interestingly enough, up to 45 minutes of cold exposure did not cause significant cell death or morphological deformation. This finding is very important because it indicates that short-term cold exposure is not cytotoxic to brain cells, meaning that therapy appears safe, at least in the short term. Cell viability remained above 90% for all points, as measured by the automated Luna-FL counter, supporting the microscopic observation. This sets the stage for us to investigate deeper molecular changes without worrying that they're just artifacts of cellular stress or death. Although general cell viability was unaffected, exposure to cold for 45 minutes did result in a visible reduction in cell size and slight compaction of the cytoplasm. You can observe here that cold exposed cells looked smaller and more condensed compared to the control group. This suggests that cold may trigger adaptive changes in cell morphology. This could be interpreted as a protective cellular response, a kind of stress adaptation mechanism. Many stressors, including cold, can cause cells to enter a more conservative, energy-saving mode without causing damage. These adaptive changes are important in triggering molecular pathways like the activation of stress-responsive genes. Now, let's move on to the most exciting part of my study. Here, you see the RT-PCR results showing CREB and BDNF gene expression levels. On the left, there's representative gel images of PCR products. On the right, there's images of quantification of expression levels normalized to GAPDH. Compared to the control, the cold-exposed cells at 45 minutes showed a notable increase in both CREB and BDNF mRNA levels. This strongly suggests that cold exposure may activate neuroplasticity-related pathways at the gene expression level. Both CREB and BDNF are well known to be involved in antidepressant effects, mood regulation, and brain adaptation to external stimuli. This result gives biological validity to anecdotal claims that cold therapy improves mental well-being. In fact, many antidepressants aim to increase CREB and BDNF levels through pharmacological pathways, whereas cold exposure seems to do it naturally and non-invasively. To conclude, cold exposure at 4 degrees Celsius for 45 minutes did not harm brain cells in the short term. It induced cell size adaptations and significantly upregulated CREB and BDNF expression, suggesting activation of neuroprotective and antidepressant-like signaling pathways. This points to the potential for cold therapy as non-invasive interventions for improving mental health, especially for conditions involving reduced neuroplasticity like depression. However, this is just the first step. In vivo studies using animal models are necessary to confirm if these changes translate to functional outcomes like improved mood or cognition. Also, the long-term effects of repeated cold exposure need to be studied carefully with a lookout for potential side effects. Ultimately, this work opens a new door to new research in the intersection of neuroscience, mental health, and environmental physiology. Thank you for your time and attention. I'd like to thank my mentor and lab team for their guidance throughout this project.